2023 Nobel laureate in medicine, Professor Katalin Karikò. Professor Karikò is a professor at the University of Szeged. Szeged. Sorry, I tried many times, but all the times.
Yeah, Z, you don't have to say
Z. Okay, Szeged.
Yes.
Okay, I have to say it another time, so maybe
Yeah.
the second time will be better. Szeged in Hungary, and adjunct professor of neurosurgery at the Perelman School of Medicine, University of Pennsylvania, where she worked for 24 years. She's former senior vice president at BioNTech in Germany, where she worked between 2013 and 22, and she co-founded RNA-RX, a company which is dedicated to developing nucleoside-modified mRNA for therapy. For her scientific achievements, she received many prestigious awards, including the Japan Prize, the Horwitz Prize, the Breakthrough Prize, and the Lasker-Debicki Clinical Medical Research Award. Professor Karikò received her PhD in biochemistry from the University of Szeged in Hungary in 1982, and for 4 decades, her research has been focusing on RNA-mediated mechanism with the ultimate goal of developing in vitro transcribed mRNA for protein therapy. She investigated RNA-mediated immune activation and co-discovered that nucleoside modifications could suppress RNA immunogenicity, widening the therapeutic potentials of mRNA. She's co-inventor of mRNA-related patents for application of non-immunogenic nucleoside-modified RNA, 18 of which have been granted by the US. Her patents on nucleoside-modified uridine in mRNA have been used to create the FDA-approved COVID-19 mRNA vaccines by BioNTech and Pfizer and Moderna to fight the pandemic. I wish to remind that the Center for Infectious Diseases at Imperial College of London calculated that vaccines prevented approximately 20 million deaths out of an estimated 32 million potential deaths that would have occurred without vaccination. For the milestone discoveries that paved the avenue to innovative disease-modifying treatments, which are now or will be soon available for many patients, Professor Karikò will be remembered, I believe, as one of the most iconic Nobel laureates in the history of medicine. But Professor Karikò is not only a pioneer of modern personalized medicine, but she's a special person with a special family. Her daughter, Susan Francia, is a 2-time Olympic gold medal in rowing with the US team. I've got the privilege to spend some time with Professor Karikò in the last weeks and actually could appreciate her humanity, depth of thought, and inspiring vision, and if I may, not only about science, but about relationship between people and life. She's been so kind to accept our invitation for a seminar today and to share her experience in an open conversation with young researchers after her seminar, which I believe will be an unforgettable experience. So please join me to warmly welcome her in our institute, and thank you very much for being here today.
Thank you very much, Jeff, and thank you, Matilda, inviting me here. And I am happy today to talk about how mRNA was developed for therapy. I was thinking about how I should start, and first I want to say about my life, but the uncertain time in these days in science, I thought that I will just talk about how discovery is born. And in academia, in a research institute, or universities, we are doing discoveries, and then when we have some kind of interesting data, then we develop it to companies, we establishing companies, and when we need more money, then we go to the large company to run clinical trial. But recently we learned that when a FDA approve a product is just not sufficient, because we need acceptance from society. And because what happened is that during the pandemic, and now we learn that this misinformation spread much faster, and here are the people who learn, the immunology in social media, and they spread different information, and we scientists has to do something. And prior to that, in this room, I talked to the journalists, because together with them, we have to do something. And today I am talking about this because you are here and you are also part of the solution. So please go out whenever you have an opportunity. I went to the local library in the US, and wherever I have an opportunity to talk to the average people, the people that they try to understand science. And we like to talk to each other, about science, but we have to talk to the public. As you heard in the US, one of the criticism was that $50 billion was spent on Drosophila research, and the people were very upset with it, because we scientists did not explain why we are doing it, because this is a model animal, not because we wanted the Drosophila live long, happy life. And if we don't explain to that it is a model animal, we discovered certain things, we have to name it. Let's say toll-like receptor, we discover in human cells has toll-like receptor. As a result of it, we could make a medicine which is not inflammatory. And that's this form, you just get this one. Thank you for your tax, the money you paid as a tax. And if we don't explain that, this will happen here in Europe also, what happened there, that, the majority of the people will be against us, because they consider us, scientists, physicians, as, the elite, and they left out from it. So we have to do, all of us. And as you know, that those who has less knowledge, they are noisier, and there is already this Dunning-Kruger effect is very well known. Somebody has less knowledge, more confident that they know the things. And as we gather more information as a scientist, then we always, have some question, maybe something different, but those people who knows less, they are more confident. But so that's, I want to say in advance, and now I talk about mRNA. So, messenger RNA was discovered in 1961, and it took 60 years when in the form of mRNA vaccine became an FDA-approved product. And every 20 years, something happened. This is the same for Cas9 and others, that scientists do discoveries, doing different things. Here in the RNA, they see that how the RNA looked. And then there are some discoveries giving a big push to the science, and then, scientists could make tests on animal, different mRNA when they could make in a tube. And finally, from 2000, new companies formed, and these new companies start clinical trial, because, those who sponsoring the companies usually want to see the result. So that's what I am talking about. So as I mentioned, the discovery of RNA happened in 1961. There were 2 papers in Nature. Okay, I was just 6 years old, so I did not read Nature at that point. But I can say, interestingly, that, 64 years ago, this paper came out. But, I heard 2 years ago, Matthew Messielson give a talk. I met Gilbert, and here, Watson, 97 years old. These people are still around. Yeah? So, working with mRNA, maybe you have a long life. And also you get some Nobel Prize, because for different reasons, Sidney Brenner get for, and Jacobs get, and Watson get for, you already know, the DNA and so on. So Gilbert for sequencing. So, if you work mRNA, likely get a Nobel Prize. Actually, I get one of mine here just to show you, just, that before you get your, before you get your own, you can check out how it will look. Yeah, so that with this discovery, that we discovered that actually the how central dogma of life is that from DNA, messenger RNA, and protein. So this RNA is coming out from the nucleus, copy part of the chromosome, and giving information to the ribosome to make the protein. So the scientists started early on when they discovered RNA. They discovered that there are a poly-A tail, and other scientists were making oligo-DT to purify it. So when they could purify, the other fellow scientists look at the other end of the RNA. So whatever you researching, it will be somebody will build on and discovering more and more. So it is always important, and please take it like that. So there is not such thing that, it is just wasting time. It is all important discoveries, and even in that year, 1975, okay, if you work on the 5' end of the RNA, you get several Nature papers. But by end of the 1975, they discovered the enzyme, which, a viral enzyme, which can do this cap, put this cap there. And if you receive the Moderna vaccine, they use this enzyme to put the cap on the mRNA. Other scientists looked at that, okay, how this mRNA translates. And of course, at that point, they couldn't synthesize. They could just isolate. And reticulocyte, the precursor of red blood cells, has a lot of beta-globin mRNA, and they purified it. And Alex Buckham in London, he discovered how to make liposome. He wrapped everything in it. Not everything, but in the laboratory, what different molecules. And a fellow scientist here, Dimitriadis, was working with RNA and put messenger RNA, which isolated and then delivered to primary mammalian cells. And he demonstrated the cells normally would not have beta globin, and on this Western blot he showed that. And I have to mention this is 1978, and single author Nature paper. Yeah, it happened a long time ago, and this was the only one figure in that one Nature paper. So in 78 it was easy, not like this. So I as mentioned, I went to University of Szeged in Hungary because I learned that this biological research center just opened there. My high school teacher told me that, the same teacher who told me that I can be a scientist. And it was a small town, 10,000 people live there, so I have never seen a scientist. I believed to the teacher, so that I can be. And there were no internet, so knowing that this research center is open. So I imagine myself, white coat, if some of you are here, somehow, I will do at the bench all of this work. And I love plants, but the only opening on this big institute was the lipid team. So I said, okay, lipid is okay, but okay, I will spend the summer working at the bench. And the head of the lipid team said that, Kati, you will have the rest of your life to work at the bench. Now just go to a field trip. At that point, I didn't know that at age of 58, I am still pouring the agarose gel, a polyacrylamide gel. So I spent a lot of time at the bench. Anyway, instead of the bench, I was collecting water fleas in the lake here. This is a fishery institute. And so that I always went with the flow. Whatever was given, then okay, then water fleas, not flowers. But then, I carried some C14 acetate in my pocket and did the first experiment. And, that was my first publication, and did these studies. And when in the fall, we went back and we started researching. Two scientists, Ernő Duda and Eva Kondorosi, came to the lipid lab, and they said that they want to make liposome because they read Dimitriadis' paper, and then they needed phospholipids. And in Hungary in 1970s, even 80s, we were behind the Iron Curtain. It was embargo, and we could not order things from Merck. And so what we did, we found an old paper how to isolate phosphatidylserine, phosphatidylethanolamine from cow brain. And since my father was butcher, and I was, not afraid of seeing, this brain things. Actually, I learned from my father, that hard work is part of life and how to make sausage. Not the Italian, we put paprika in it, yeah. So, you need always to get a plan B if, my science career is not working out. Anyway, so we did and delivered, the DNA in that time, with this liposome to the cells. And then one day, head of the RNA lab came to the lipid lab. I was working there, and Janu told me that, do I want to do my PhD in RNA? And since nobody invited me anywhere else, I said yes. Again, just I want to show you that, I am not a visionary. I have no special talent, no special memory, and not a visionary to figure out that lipids, RNA, and one day I need that. I just went to the flow, with the flow. And what he wanted me to synthesize this 2', 5' oligoadenylate. It was just discovered the prior year. We are now 78, and so this was, try to synthesize. And he told me, he's an organic chemist, he told me, just set up a viral screening lab. It was easier to say than do, but that's what I had. And I worked together with the János Ludwig, a fellow graduate student, why the head of the lab went to sabbatical to the US. So that's what happened when two students start to work together, and you have to be always nice to fellow students, because even today I ask advice from János, all of the organic chemist questions. So this was this 2', 5' oligoadenylate, and it is a natural molecule. I always stay with naturals, and it is oligoadenylate synthase is making from ATP, and this molecule, this short RNA molecule, can bind to the RNase, the latent RNase, and then it can dimerize and cut up the viral RNA. That was the idea, and that was considered that part of the interferon mechanism. And so this is now we are in 1985. I am 30 years old, and some pharmaceutical companies sponsored our research, but because we couldn't make it delivery even with liposome or calcium phosphate co-precipitation and many other techniques, it was not feasible for human application. So the company said that no more money, and then I had to leave Hungary. I have to say, I never wanted to go to America. Try to first find a job in Europe, but again, behind the Iron Curtain, we were not eligible to get grants and something. So anyway, at age 30, here I am in United States, and in Professor Suhadolnik's lab, who did the basic book on modified nucleosides, and we did the different 2', 5' oligoadenylate, some antiviral compound, because we needed so badly. And it was already known that double-stranded RNA can induce interferon. Interferon was discovered in 1957. 63, we already knew that double-stranded RNA can induce. This interferes with the viral replication. Not until 2001 we knew how. But anyway, we try to help HIV patients, because now we are 86, and that's a major problem in the US. So we did this double-stranded RNA poly-IC was very toxic. They tried in France already, and the therapeutic window was very narrow. So we did a mismatch double-stranded RNA. Later, after 2000-something, they showed that it specifically activates TOL3. So we treated patients quite a high dose, 10 HIV-infected patients we treated, but unfortunately their interferon system is already compromised by the virus. But as you can already notice, I made small RNA, I made long RNA, made double-stranded RNA, always about RNA. And meanwhile, the field of mRNA was moving ahead. And 1984, scientists discovered that how they can make in a glass tube, how they can make mRNA what they want. So they don't need to isolate from reticulocytes. And these were scientists from Harvard University. And they tested out here I just reference some papers here. They look at bacterial RNA polymerase, phage RNA polymerase, and different RNA polymerases, and they found that actually the phage RNA polymerase has high fidelity, high yield, and that was the best, and the promoter is very short, and they created plasmid. They provided the basic 4 nucleotides and the enzyme, and the template was this plasmid, and then actually the human beta interferon RNA, what they synthesized, and they get some capping enzyme, and when they injected this to frog oocyte, I have to say frog oocyte, the tissue culture cells were not round at the beginning. So if you go to a genetic lab or something, you always see a terrarium with frogs in it, and they collected eggs because the eggs were large and they could easy to inject. So when they injected this messenger RNA, which had cap poly-A tail and coded for beta interferon, to the frog oocyte, incubated overnight, the medium has antiviral effect. And so this gave a big push to the field. I have to say that in the 80s, the mid-80s, the scientists were so helping each other. It was I remember when I sent a letter, letter in that kind, no internet, no email. It was a letter with stamps on it to Paul Krieg, and I asked him, can you send me this plasmid with this promoter? And he sent me on a Wattmann paper, and he even said another one. He said, I have even a better one with T7, try that. And yeah, so hopefully the Intellectual Property Office, nobody is here today to complain about that we shouldn't do, but it was, everybody tried to help the fellow scientists. And after discovering, you would see that in the 90s, more and more publication out using messenger RNA. Scientists demonstrated that injecting messenger RNA coding for luciferase to the muscle, it translates, and then applications were here. Floyd Bloom, who was the editor of Science at that point from Scripps, they injected vasopressin mRNA to the brain of the rat, and it was cured. And other, made influenza vaccine already, 1993. And the cancer vaccine is also 95. And actually, Eli Gilboa at Duke University, he took the cancer vaccine because he established a company, Argos, and they did a human trial up to phase 3. And those patients, they took cells out, they generated dendritic cells ex vivo, treated with RNA, put it back to the patient, but unfortunately it was not successful. So who has to read a lot of paper appreciate that just every year there were one paper there, and it was easy to follow the literature. And so I thought I didn't understand why these scientists, Floyd Bloom, on this paper, every authors I followed for even today. They never published again mRNA. And I thought that because, small amount of protein was made a short period of time. But now that I became more known, I met some of these people, and I learned from them that they didn't get funding for their grant, and that's why they stopped the research. I didn't know that, and so I was applying for a position to University of Pennsylvania. From Temple University, I was kicked out again, but not because I was doing anything wrong. It was just Professor Suhadolnik wanted me to stay there forever, and I got a job offer at Johns Hopkins. I wanted to go there, but in the CV you didn't mention because he called them up and told them that I am a fugitive and shouldn't hire me, and they withdrew the position, but they wanted to make it up. So if I wouldn't be here right now, I would be honorary doctorate at Johns Hopkins. They want to try to make it up. But anyway, I got a position in Eliot Barnett lab, and it was a faculty at University of Pennsylvania. So I supposed to get my own funding, and Eliot supported my salary for 7 years. I still couldn't get any money, but Eliot was just 8 days younger than me, and I convinced him that mRNA is the future, and he was interested in urokinase receptors. And this protein is highly modified, glycosylated, GPI linked, and so on. And what was the most surprising when I cloned and made the mRNA and we delivered to a mammalian cells, that the cells knew how to decorate and they made functional protein. So we immediately thought that we will use this technology maybe for cell therapy and maybe, try to find a disease, and that was like bone marrow transplantation, that we can extend the tip of the chromosome if we deliver telomerase, and then maybe older people can give bone marrow. And because he was working, bypass surgeries they did, we also did testing blood vessels, and I dripped through mRNA, and 15 minutes later I could measure the protein which coded by this mRNA. So it was feasible that during bypass surgery, they have in their hands the blood vessels and things like that. And so with Elliot, we did with a lot of interesting things, but he stand up for me because in meantime, University of Pennsylvania demoted me from faculty position. But there was a medical student in the lab, David Langer, who by that time, when Elliot was leaving, because he couldn't be tenured, David became a neurosurgery resident, and he convinced the chairman of neurosurgery that neurosurgery department needs a molecular biology. It was 1997. Even today you are laughing, yeah, that what she should do there. But so be nice always to the medical students, in the lab, because thanks to him, 17 years I worked. This was my laboratory here, and with low budget, you see no lab coat, other things. That's me there, and so we try to develop mRNA therapy for a patient who suffering subarachnoid hemorrhage, and they have major blood vessel constriction. And so we come up with the idea that maybe nitric oxide synthase mRNA would be good, and he said that he could reach that major blood vessel. I made the mRNA. I showed that it can make hanno, and everything is working, and did studies. But he had to leave, but thanks to him, every year he came back when they tried to terminate me, and for at least 17 years he could reach that I stayed. And I met Drew Dreissman at the Xerox machine because 1997 we did not get the publication digitally. We still copied and put to the file, read it and put to the file cabinet and so on. And with Drew, on the Xerox machine, I brag about, that I do RNA because of already 10 years I was working RNA, and Drew is a quiet guy. According to his wife, he has a limited number of words he can say a day. And his wife said that he wanted to know what research is going on. By the time he goes home, he used all. And so it was, with different personality, with different knowledge. I did not know anything about, immunology, because what I learned in school, it was already outdated. And I learned from Drew, and he learned the RNA from me, and then we, work together again. So invention can come from a big team is working on a project, a problem, or maybe just two person, like I work with David or I work with Drew, that we knew different things and we respect each other, work together, and discover things. So that he wanted to make, vaccine for HIV. And, I want to make mRNA code for therapeutic protein. So what we did, the first experiment, we discovered that, the messenger RNA induced TNF-alpha, which is, inflammatory. And Drew thought that it will be okay still for vaccine, but, for me, for treating stroke patient would not be a good idea. And so we asked the question, is it the RNA because it's coming from outside to the immune cells, these dendritic cells, these human dendritic cells Drew generated, or maybe some RNA is not, some other type of RNA is not inflammatory. So I had I already worked 10 years on RNA, and then I had a collection of different isolates from tRNA, ribosomal RNA, whatnot. And this purple here is the in vitro transcribe, which I made in the tube. And surprise, surprise, these were less inflammatory. And one of the RNA that was transfer RNA was not inflammatory at all. No TNF-alpha was induced. And so, we thought about many things. The tRNA is shorter and different ends it has at the 5', 3', but it is also that 25% of the nucleosides are modified. And that came the idea that maybe modification of the nucleosides makes RNA non-inflammatory. So Drew just said that, okay, I should make the modified RNA. Yeah, but which one? There are more than 100 is known, and I didn't know that we need all or some or which one. And because most of the, we make the mRNA from the 4 basic nucleotides, and enzymes put all of these modifications on different position there. But, what are those enzymes? So it was not easy just to make one. And I knew that 2 modification was known. We are in 2000 about, or 2001. Because Tamás Kis, who was fellow student in University of Szeged, we lived in the same dormitory, later work in the same institute in Hungary. So he discovered 2' methylation of the ribosomal RNA and pseudourylation of the ribosomal RNA, and he discovered the SNO RNA, actually a guide RNA, show that where the modification has to be. So I called him up and I said, Tamás, send me stuff. I have to modify the RNA. And that time it was, it wouldn't work in tube and so in a test tube, and so it was not applicable. So I had to call up János Ludwig, who we were student together in Thomas Jenő lab, and I asked him that, okay, I have to make RNA, modified RNA differently. Not as nature does, because Tamás couldn't have, but maybe I will purchase nucleoside triphosphates, which already the nucleoside is modified, and try to, cross my fingers that the phage polymerase will incorporate. I wanted just only use the natural one, and then I purchased 10 that was no more available, and it turned out that 5 of them incorporated to the RNA. And that's how I made the nucleoside modified RNA. And then we tested out, Drew made, actually he isolated from the blood human dendritic cells, and we tested out. And what we found is that some RNA was still inflammatory, TNF-alpha was generated, but some of the RNA, we found it was no inflammation. And when we looked closer, we found that every time the uridine was modified, it was this RNA caused no inflammation. So we said, oh, we didn't know that, but actually the next week when we opened the Science, there was a publication. Yeah, this is what usually happens. And they said that, poly-U can activate toll-like receptor 7. Yeah, but we knew that how to modify not to activate. So at least we have at some edge. And of course, Drew wanted to make a vaccine. I want to make therapeutic protein coding RNA. The translation was important. And when we translated in different cell type here in 293 cells, what happened is that we found that pseudouridine containing RNA, we get 10 times more protein. Even when primary cells, human dendritic cells, we get 10 times more. So we spent 2 years figuring out why. And of course, some uridine modified one didn't translate it at all. What happened usually in the United States at that point, you file a patent. As I mentioned, it's important that previously here that, the patent filing, it costs money. So when we approached the department there at Penn, the officer asked that what is this useful for? And because he happened not to have hair, I said maybe hair growth is good, mRNA. And so the patent was filed. And after that, we also established a company because it is important. Nobody will walk University of Pennsylvania IP office and say, oh, pseudouridine, that sounds great. Nobody. We knew that it is important, and with Drew, we established the company RNARX, but nobody is nodding that we know about that because we didn't get our own patent for our own company. So what we did, we wrote a business grant. Because, you know, I was already demoted and mistreated there at Penn. I was so much ready to go out the door. And, but, you know, we get the money. And that was the total all in my life in US I received. This was $1 million from the taxpayers' money, and it was like four or 5 years paid my salary. I earned, 2010 I was like 60,000. I went there, it was 40,000. So in 20 years, you know, I get 20,000 increase, but you know, the healthcare and the parking space exponentially increased, so I get less and less money, but it doesn't matter. You know, I didn't need any hobbies. You know, I spent all weekends at the lab having fun there. Anyway, so we promised that we will use pseudouridine containing RNA to treat anemic animals, and eventually we can treat people. And so we did, if somebody works with RNA or DNA, you know that how small amount is 0.1 microgram. 0.1 microgram pseudouridine containing erythropoietin coding RNA when injected to the mice. Even 4 days later, we could detect the protein, which has half-life is 2 hours. And it was physiologically functional because hematocrit increased. And when weekly injected, the animals, you know, that increase was maintained. And the RNA was not causing any cytokine increase. So this is 2012, and it seems everything's wonderful. That's what my daughter got the second gold medal. You know, one she got in Beijing in rowing, and here we are, my husband and Susan in London. But I was told by university that I have to retire. That was, I was 58 years old, and believe it or not, you know, I felt that I am in the top of my game because I get 2 job offers, one from BioNTech, other is Moderna, vice president. Yeah. But it was not in this fancy building, we were just on the campus. And, you know, when the company is a handful of people, you know, you walk in, you can be a vice president. Anyway, I selected to go to Germany and at BioNTech, and we were not here in this nice building yet. And we were further improve the messenger RNA, and many things what I did in University of Pennsylvania, we had to make it scalable, industrial technology to develop, and we optimized the codon, we purified the RNA, all of them is improved the translation, and eventually, not a small amount of protein, short time, but this extended time we could detect the mRNA was there, and more protein was produced. And all of these things, you know, is not just happening by us, but all of this improvement was in many layers. One of them is the genes. You know, at the beginning, I had to beg people to send me what genes they cloned, and I was interested to make mRNA. And then later on came the PCR. I mean, 1989 was, tech polymerase was the molecule of the year. So young one think that all of these things were around forever. No, 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 it was not. RT-PCR, TA cloning, what I did most of the work, and later we could order genes, but those are already well defined, like Origin and others, we could order. And eventually, from 2000, some companies started to make a gene for order. And from 2006, I labeled here, this was the first time it was affordable, because when it was $4 per nucleotides, I couldn't order a genes. But from 2006, it was affordable. And improving also on the RNA. When it was, CAP was discovered, pharmacy already, you could purchase from pharmacy. CAP analog, polymerase, and capping enzymes were available, different companies, and we could have enzymatic capping, and we could incorporate co-transcriptionally cap into the mRNA, and also we could incorporate nucleosides, modified nucleosides, analogs. And of course, the other thing was the delivery, because the RNA had to put in some protective material, which could be a liposome, or commercially available lipofectin or transit, and protamine, which is present in the sperm DNA, and lipid nanoparticle was introduced in 2013 when I first heard about lipid nanoparticle, and then with RNA. And in addition to all of what we did in the laboratory, I have to mention that many things advanced. Because when I went to the United States in 1985, the biggest advantage was that I could listen to lectures. And really, we were sitting on the floor and then going to other city, for example, driving there, because Gallo is saying something about HIV. There were no internet, no nothing. The only way we could get information to go and listen. And you see the PubMed, you think it is forever? 96. That's what introduced, or clinicaltrial.gov, yeah, and YouTube from 2005. So I made this just to show you that not everything was around forever. But I was talking about here RNA and why it is important, because right now many drug which is protein, and the protein is actually quite expensive. So if we would replace the mRNA with mRNA, and instead of recombinant protein or purified protein, the patient could make their own drug and translate. And even if eventually the drug is protein, screening, testing out with mRNA is much faster. So it makes affordable. That's very important for Drew and me. It was very important that we will make a medicine which is affordable, not like $1 million value. And if in these days you hear about RNA, it could be RNA could be linear, which some of them is conventional. For cancer vaccine, for example, you need cellular immunity, and those are conventional RNA is used, at least for BioNTech. Others use also modified one for that. But for example, for replacement therapy or vaccines, all of this is nucleoside modified. And there are replicative RNA also was already actually approved in Japan as a booster vaccine. And in these days also use circular RNA, that's internal ribosome entry site. At the beginning they used actually, this is considered a novel technology. In 1994 I already had grant rejected for circular RNA, but for those who know, everything is new. And there is also cap dependent, and which can be covalently linked or can be trans. And if we consider what is today is more than 150 human therapies, clinical therapies ongoing in the whole wide world, and 50%, about 50% of those for different kind of viral vaccine development. Norovirus is already phase 3, and bacteria, tuberculosis and Borrelia, which Borrelia causes Lyme disease, and parasite like malaria. And for cancer, there are FixVac, which is shared tumor antigen targeting vaccines, or there are individualized one. But I participated in BioNTech is when mRNA code for cytokines, and that we use for head and neck cancer, and went with Sanofi in clinical trial injecting head cancer, which is melanoma or head and neck, other kind of cancer, and make the cold tumor hot. So the immune cells will rush there and recognize the problem. And there are also, I participated in generating bispecific antibodies based on mRNA and conventional antibodies. And for acute disease, actually, Moderna use VEGF-A mRNA. VEGF generate new blood vessels, the protein, and this encoding mRNA was injected to the heart in patient who had heart failure, and they also treated necrotic wound of patient who has diabetes and couldn't heal their wound. And for genetic disease is a more application there for cystic fibrosis, for sickle cell anemia, and others, and also gene therapies. And autoimmune disease is myasthenia gravis is phase 3 by Cartesian. This is CAR T cells, but using messenger RNA to generate. And not only the human, but animals is benefiting messenger RNA vaccine. Elephants was injected, not too big dose, 150 microgram for herpes virus, and horses were injected already. I don't know, the shrimp is a little bit challenging, but they injected also ducks in France, and it was a replicative RNA. And what is left for me is actually, inspire the next generation of scientists, the young one. I did not write these books. They wrote about me. You can see Dreissman and me thinking about and doing our research, and also I wrote myself in English this book, and it was published here in Italy and many other different languages from Asia, and is coming out in Iran and Serbia. And my book was 10 days ago was selected by the Italian Premio Asimov Prize for high school students, and almost 15,000 students participated, and of course they are the real winners because they were reading all of these 5 books, which was recommended by professors and students from universities. And when usually I get an award, I usually say thanks to all of the people, even those who try to make my life difficult, because made me work harder, and I'm more resilient. But when I get the Nobel Prize, actually, I didn't invite those. Only I invited my husband, my daughter, my sister. These are the people who I did the lipid work together. These are my teachers from University of Szeged, teaching me nucleotide chemistry, genetics. And here is David Langer, who made sure that 17 years I have neurosurgery position, and Elliot, who hired me, and he had to leave because he stand up for me. And there are some young ones from the city I worked and graduated. And Gabor is here today also. He was there in the ceremony. And we work together on a project, which I will talk about more when next time I will be back, that what I am doing here, besides talking to you, why I am here. And that would be all. Thank you very much for your attention. I usually say to the young one, when you want to be in the spotlight, that you should be an actor or singer, not a scientist. The scientist is rarely there, and get uncomfortable.
Thank you. Actually, thank you so much for sharing with us this fantastic journey that, but somehow, if I may, reminds me something that Charlie Chaplin said, which is that life looks like a tragedy in close-up, but becomes a comedy on a long shot. And actually, this is what it seems to be, how I would like to translate your, I don't know, resilience or whatever you like to call it, but it was really an incredible journey through, and really appreciate how you jump, as you told us, how you jumped from one tough thing to another one, but eventually, maybe it's a good way to get this this prize. So, Professor Karikò is here. I'm sure you have a number of questions, and please, who want to start? Please.
Well, thank you very much. I'm really emozionata. It was really inspiring. I was very touched by the first diapo when you show the role of the scientist. You work in academics, you do some discovery, and then you have to go to some companies. But you put the last, I would say, at the end of the last role of the scientist is to explain the job of the scientist to the public. Can you say something more about the how do you say in English, the education role of the scientist?
Yeah, so that's what I ask you. Yeah, that and everybody who is here, that we have to find a language to communicate to the average person. First you start family members. Okay, I know once my daughter was 8 years old, and my husband who is a mechanic. I told them, you have to sit down, listen. That was the circular RNA. I get this idea, and I explained to them that this is it. Because I tried to explain this discovery, and recently they asked my daughter, what was your reflection when your mom told? And what did you ask something? And she said, at the end, I asked, can we have dinner now? So that, but because we try, and we should try in the family members, if they don't understand what I am doing or you are doing as a researcher, then you have to keep trying. Recently, the Stanford University asked all of the graduates, they have 3 minutes to tell what they are doing, why it is important. 3 minutes that anybody could understand. And they cannot use the complex languages, because then, 3 minutes is up. So that we have to practice. Because if we don't say there will be those others will fill the gap, and then they will say stupid thing, and we cannot afford that. Because if we don't educate them, then we will, eventually go down on their level, because they see us as an elite. So I don't know, you have to figure out. I know that it is difficult when Drew also said that the RNA is like a car, it has 4 wheels and we change one, So, yeah. But something we have to say on some analog things, whatever you do.
Thank you very much for this super insightful presentation. I have one question about the delivery of messenger, because you show us that just in the beginning you develop your way just to deliver messenger, and after you use some commercially available products. So which is the reason why of that choice, and which is the importance of the vehicle just to deliver nucleic acid?
Yeah, so, at the beginning, everything was about what you can reach. So I have to say, as my daughter is a rower, if you have a rower relatives, that they have always blisters. So I punctured each of them, that was my job. And I was thinking always that a little collagen 7 mRNA would be useful, but, I had to clone it and so on. Anyway, so we thought about wounds, because if you deliver a protein to a wound, it will wash away because, with time. But if the cell would pick up there the RNA, then they can constantly secrete. And so that was first localized. And then, come the IV delivery, and almost everything ended up in the liver, And then, they focused on liver diseases. And now that they can decorate, some people put antibodies to the wrapping material, which is different lipids or so on the RNA, and then they put antibody or some protein which binds certain receptors, and they can target specific cell type. or specific area. And some of them just changing the composition of the lipid and then will end up based on charge or other things. And they check it out if they IV inject where it will end up. So there are a lot of researches going on that. But right now, they can reach bone marrow cells. So you don't have to take it out, but you can deliver mRNA, and there is clinical trial is ongoing. They also, for CAR T cells, which is, they took out the cells and T cells and they delivered virus and so on. But now that they deliver an mRNA, which will target specifically on T cells, and when enter, it will express the CAR on those cells. So they don't need the expensive cell therapy. It will be just injection of the mRNA. And a lot of companies and form on that to making more specific the therapy. So less side effect and more specific. So targeted delivery.
Thank you very much. Thank you.
Thank you for sharing with us this beautiful journey. You highlighted that at the beginning of your professional career, one of the main difficulties was how difficult it was to access to the information and to the data published at the time. Now we have the opposite problem. We are surrounded by so many information, so many papers. So what would be your advice to a young researcher to navigate in this sea of millions of papers and PubMed and so on?
Yeah, so at the beginning, actually, I made an Excel sheet, and when I checked out, never miss a Nature or Science, and was reading A to Z. I learned a lot, by the way, but anyway, and then what I did now is that I set up alerts, like PubMed alerts, for scientists who are doing something that I want to know that what they are doing, or certain words, and if I get hundreds of publication, then I had to refine it to make sure that I get those which I really wanted, and Google alerts so that people what they are doing. Now I started to work at BioNTech at 2013. Then I started to pay for like endpoint news, stat news, and then reading every day that what the competitors' companies are doing, what is their goal, how they measure, why they fail, that we don't repeat that. So to be to know the picture, what is going on our field of mRNA, and then I had to pay for those, but even today, every morning I just check what's going on, and so that the difficulties now is that too many information and how you filter. And when I was reading a publication like the mRNA, now that for let's say generating pluripotent cells or using RNA on different field, I start to open tables. I love tables, Excel tables. And then so that I put there that, I thought that is important for me to know. And then when I wrote a review, I just look at, or I'm reading a paper, I said, oh, I don't remember that. I look at my table, oh, that is missing. So insert a line, and then I put it there. So that's, and then I started to do the same thing for when I do the research, so that we did a lot of animal experiments, for example, at BioNTech, and we used different RNA on different cap, different formulation. We measured interferon, we measured EPO, erythropoiesis, many different things. And then I realized when somebody asked, have you ever measured so we made the table. And it is easier if you do the beginning. And then you can put internally link to a page. And then when Ugur, let's say, asks whether we have ever done with that formulation and tarara, and then I just click on it, and then I said that was the result. And he was very impressed. Although, Germany, they are very precise, but it was still something. So that had to build this kind of thing. And finally, I realized, okay, we are talking this people, and then I had to make for people, to who they are, and then they left or go. And so, because I couldn't keep in my mind. And Giuseppe already knows that the computer has to be nearby, like here, because whatever is not put here, you know, it is there. And so quickly I can take out wherever I am, yeah. But it's difficult.
This is exactly what she's done 5 times today. This morning, actually, she's opened her Mac and wrote something. We don't know what she did, but very likely we'll see it in one of the next Nature papers. I don't know, actually. Any other question? Okay, Francesco. Oh, closer. Where are you?
Okay. So thank you very much for this inspiring presentation. So I have 2 questions, one personal and the other scientific, if I can. So I would like to know how the Nobel Prize has changed your personal and professional life.
I have to say that before the Nobel Prize, for 3 years, I get a lot of awards. 40 years, I get nothing, you know. In the United States, if you have an R01 grant, means you are a scientist. I did never get an R01. And so it was, for me, all of a sudden, you know, to the spotlight was like, in 2020, you know, December, CNN called me, you know, and I, online or you know, live, I have to say something, you know. I mean, I was terrified that that's the other thing, we are not prepared, you know, to talk. Yeah, the, you know, reporter, and you have a responsibility that not say stupid thing or something that people might misunderstand. Yeah, so there's, yeah, so it was a lot of pressure for 3 years, and you know, and all of this award is, you don't think that you go, you know, just here, we dress up and go, and thank you very much, and go home. No, it is like 10 days, and you go out, and every hours, you know, give interviews, talk to the children, talk to the public, and so all of this award is like that. So it's very stressful. So be prepared. Prepared. And so it's not easy, and so it, of course, everybody knows about Nobel Prize, and nobody said that, oh, Kati, you get the John Scott Award. This is the oldest American award. Wright brothers, Tesla, Marconi, Edison. You don't know, because no royals in the U.S. Now maybe we will have one, but you know, when the royals gives you it, something is different. And when you have a big ceremony or something, and when you just go there and thank you, that you know, the award is not as known. But I mean, I realize getting an award, it means that it's not about me. It is about all you know, all scientists. Every time I say thanks to the name of thousands of scientists who work diligently. When it was about Corona, I always said that the real heroes are you, the physician. I never risked my life. You did, you know, all of this, whether it is a nurse or a physician, or even the cleaning lady went to the room where somebody was infected and you had nothing to protect yourself, and you went home or tried to protect your own family. And you did. And all of those people who were volunteering for vaccine, they said, okay, give it to me. You know, it was nobody get that before. So they are the real heroes. I was just playing, as my husband said every time when I went to work at the weekend, that I am just playing there. So it is always realizing that it is not about me, and when there is an award, it is the attention is on science. And that's, we rarely have that, you know, because the journals are writing about those, I don't know, Kardashian who did, I don't know what, but, you know, that's what the people. And they are not writing about us, about you, that what you are doing, how you're saving life of the people. And then we have to, you know, keep this going. So I went out every time they asked me to say something. And I not just, you know, mentioned people who try to make my life difficult, but I also mentioned them and mentioned that so many people worked on. And then you, the physician here, worked hard to save lives.
Thank you very much. So the other question is related to gene therapy approach for rare genetic disorders. So in comparison to mRNA approach, how do you see the AAV-based approach in terms of, cost, efficiency, applicability, and safety?
Yes, you heard about the little boy in the Children's Hospital, KGJ, and there was a messenger RNA was delivered, and quickly identifying the errors and could fix it, and try to do that with the AAV or any virus. Creating takes longer time, and they could create so many tools just to prove that it is doable. So that's the answer already, that mRNA is quick and more affordable and can be used. And yeah. Anybody?
Thank you very much.
Francesco.
So thank you very much, Dr. Karikò, for this very thoughtful and presentation. You conveyed so many messages to us today that I will ask Giuseppe to get the recording of this lecture, because I want to listen and re-listen again to what you said. Among those messages, for instance, I think the most important is the resilience. It's a message about resilience, and it takes a lot of resilience in order to do research. So thank you for that. But let me get to the question. In this institute, we operate the largest volume of gliomas in Italy, and probably even in Europe, we are among the first. So what would be your vision about the treatment of gliomas based on the technology that you have developed? And let me ask you also if there is the possibility to do something together. Thank you.
Yeah. So actually, in Florida, I mentioned glioblastoma was used clinical trial messenger RNA-based vaccination, and I am not. When I went to BioNTech, it is an immuno-oncology company. I work in protein replacement. I am not a cancer expert. I am not even a vaccine expert. That's Dreissman wanted. Okay, I learned things there, but I am not an expert on things. I try to be expert on something which I can work with Giuseppe and some others. And so technology is developing, and a messenger RNA, if you want. In Berlin, in November, there is a mRNA therapy. If people would come there, and from 2013, every year, Berlin, Boston, so they would know that actually even vaccines were already mRNA-based vaccine with lipid nanoparticle was already in human trial prior to 2020. But the people were not there, and they thought that this is the first time when we use for corona. Moderna did already human trial for bird flu, Zika, CMV. So that many things you can learn if you come, and there are experts who are using glioblastoma vaccines in human trial in University of Florida. Yeah. Published last year, 24.
Other questions?
Yeah. This, resilience is coming. What you mentioned is when I was 16. Not question, but I am answering. When I was 16 years old, in high school, the teacher, we sent out letters to Hungarian, because we couldn't speak English, yeah? I learned Russian and Latin. That was important that time. And then to János Seje, who coined the word stress. And he wrote a book, and because he was Hungarian, it was translated to Hungarian. And this was about how to handle stress. And I can tell you, I would not sit here and wouldn't get that if I would not read at age 16 the book. And it was about that he showed that the stress can kill you, but not because the stress, but how you perceive. And so we practiced there, you know, very simple way in the school, in high school. So it was like a simple thing, you know. Yeah, I say something to Giuseppe and hurtful, and then I regret people how they react, and I say I shouldn't tell and bothers me, but then I think yeah he started and he you know that's how people, and then they spent, and what Shea said, wasting time with this. He said that if you feel that way, you go there and say, Giuseppe, forgive me. And that point, it says also, doesn't matter he forgive me or not. Do not let your own feeling depend on somebody's decision. I did my part, and then I learned next time I don't say quickly something. And when I go there, I don't say that, please forgive me, Giuseppe, but actually you started, you know, not. Because, and also
she's kidding.
Yeah, so that it says also that the people like to angry and revenge. And he said, you never do anything because it escalates and you never can calm down. When you want to, you know, slash the tire of somebody, you know, the boss, because not promoted you or something, you know, you have to think about how you can be grateful to the same person you want to just do some revenge. And he said, because it calms you down. And if you are grateful, it is always much better than to be angry. And many people are angry with somebody and don't even remember later why I was so mad, but you know, they are keeping that. And we practiced there in high school. And then it was, you know, if you read the book, you know, at the beginning, one of my other high school teacher, you know, wanted actually told me that he will make sure that I will not be accepted at the university. Yeah, I was valedictorian at elementary school. I was 8th grade. I was 3rd best in the country in biology. I was, you know, very active, and he said that, you know, he knows somebody and he will make sure that I will not accept it. And so that, again, as somebody once says, so what is my reaction? I can say, oh, okay, I don't go there because he already says something. You know, in the 70s in Hungary, somebody said something, they can do that. Or I say, okay, I will learn so much, I will spend all summer reading, studying, that I will be the best, and they have to accept me. So it is always your decision. And that's what, you know, I don't realize, you know, or didn't realize at that point when he said, I didn't say, oh, thank you very much, now I will work so hard. But, you know, that's what. It is your decision. Or whenever they kick me out, I mean, University of Pennsylvania, Ivy League school, I was 58, and they said forced to retire because I didn't bring in the money. Now they don't complain because of the $2 billion they collect for that patent, which, you know, finally they filed. But, you know, again, if you spend your time feeling sorry, because that's what we like to say, oh, tell the colleagues, you know, isn't that, you know, why they kicking me out, you know, I work all weekends, and you start to feel sorry for yourself, and your friend is saying the same, and you just waste your time. And that you have to say, okay, somebody made a decision. I tell the young one, don't let others define you. You just think, what next? What you can do. Okay, I came to Germany, okay? So that's what you have to do, not to maybe tomorrow you get kicked out and be happy, oh, Kati said that, okay, I just have to focus the next one. But that's what, you know, then you are immediately, I have to do this and that, and that's what the same. You get your publication or your grant, you submit, and you read you know, the criticism coming back, you automatically say, oh, they are stupid, they don't understand, you know, and you get mad. If you conclude that, you don't have to do anything because they are stupid. You cannot change that. But if you say, okay, they did not understand, that's why. Then you can say, okay, I have to write it better. Maybe I have to ask somebody, read it, and immediately you can see that what you can do. This is, I know that it is a little bit painful, but you know that it makes always that you can act. And if you live like that, everything can happen the same way around you, but you live a happier life. That's what it is. So when I was working at Penn, you know, that I worried more about, you know, the Schuylkill Expressway traffic jam like we have this morning. Then who sits in the White House? You know, it didn't influence me that much, even today not. And so it is so that you can always, if you say, okay, I was late in the meeting, like, because the bus just left and I missed that. You know, when you say things like that, nothing to do, you're the boss, it is the boss. But if you say, I didn't leave early enough, then you know that next time I will leave earlier. Immediately something to do and you learn. And if somebody say bad things to you, let's say now, Giuseppe would, you know, I would learn that, okay, this is not nice, I will not say such thing another person. So I learned. And so everything you can learn. And okay, I learned. And that's it. So it's a Stoic philosophy. You guys figured out, and you know, Marcus Aurelius helped you, and that's that you are focusing on things what you can change, and don't think about things that you have no influence. And that doesn't matter what happening other corner of the world. You cannot do anything, and just discussing is just wasting time or thinking that, oh, I should have done something. That was always Shaya said, that people are saying, oh, what if? He said, when if you are here, stop, come back, because you already made a decision. Okay. That's enough.
Thank you. Thank you. Let me make a question, actually, which is on a completely different topic. Actually, you mentioned a number of times the need to have affordable medicine, so low cost for medicine. And in Italy, we have a public healthcare system. This is a 100% public research hospital, and this is a hub for a number of genetic diseases. And actually, at the end of the day, actually here we are treating a huge number of SMA patients, of TTR amyloid, and many others. But actually, still the cost of the drugs is pretty high. So, of course, it would be great, and in our country, actually, where we can do just negotiating a bit, not that much. But what is, in your opinion, the trajectory that actually maybe other countries could undertake to promote a different culture in the development of drugs? Because the focus on rare disease is here, and clearly the story of the kid that has been published in the New England Journal of Medicine tells that we can, from a technological perspective, move towards a personalized medicine, but still the cost of the drug remains pretty high. So what has to happen, and what is the role of the scientific community along with that of the industry and the governments, I guess?
Yeah, it is difficult to answer, because there are different countries, how they handle. Like I heard, like Indonesia, there is a child who has brain cancer. They don't do anything because they cannot afford, and it would cost so much. And then the same money they would spend some kids that, they can be normal and whatnot, and that's different countries, depending on their financial situation, make a decision that, I'm sorry, this happened and we cannot have. Other drugs which might, like for cystic fibrosis or sickle cell anemia, they said, okay, $3 million per person, because that's how much it costs if a transfusion and tararam, and they can say if the drug is below that, then it is can be affordable. And the technology that try to develop, I mean, let's say just sickle cell anemia. That, I mean, there were Bluebird Bio tried to make the gene is not good, so we deliver a gene which is correct. And they start to work, and it was, doing very well. And then all of a sudden, they discovered that BCL11A is the transcription factor when we're born, and now we are not needing the alpha gamma, but we need the alpha beta for the hemoglobin. And then we switch from gamma to beta, but the beta has mistake in it. And then they discovered, okay, if we destroy this BC11A, then you are fixing the patient. So people start to develop Cas9 for that interrupt. And then they couldn't even finish or go to the to market. Already others just not cutting with Cas9, just changing one nucleotide and the aminase, and then they get the same result, that the patient now would make the gamma. And so it is, they cannot recover the investment of those companies because they couldn't even go to the market. Already they are, a more advanced company, a more advanced technology is pushing them out. And it's not really answering your question. I just want to say that, when some drug is so expensive, is just so much was spent on money on developing it. And so I find it, this $1 million drugs, some of them, they are withdraw the paying for the license because they realize that nobody can afford. And how you can say for the taxpayers that you have to pay so much money to save that one child, which might good or not good. So it is, it is ethical, and you know better than me.
But if let's say a gene therapy would cost 1.5 million per person, mm-hmm. What is in your opinion the markup on that? So it is really that, because 1.5
if it would
million
save a person, and then that
now of course if you save a person, the cost of a life is I mean, it's costless. Actually, it's not a matter of that. But the point is that if eventually a drug will be 1.5 million per person, which is 15 million per 10 person, and then so on, whatever is the number of patients with rare disease, if though it is pretty rare, will be more than a dozen. So the cost at the end, when you have more patients, should at a certain point
I don't know. I'm not
ethicist.
You guys.
No, I don't know.
Matilde, you are the ethicist.
Matilde has a question. Well, more than one.
What I will do now is that I will read some pieces of your book in Italian, and I will do the questions related to it.
Oh.
And I will start with the one word, kutatò. Kutatò in ungherese vuol dire sei una che cerca. Ed era come il suo padre chiamava Katalin e diceva: mio padre voleva dire che stavo cercando delle risposte, delle cose che ancora non si conoscevano. Voleva dire che stavo cercando la verità, o perlomeno quello che un essere umano può trovare nell'arco di una vita. Intendeva anche dire che il mio scopo era la ricerca in sé per sé, non si sbagliava. Nella ricerca di base, che è ciò che ho fatto per gran parte della mia vita, stai per definizione facendo qualcosa che non è mai stato fatto. Non esiste un modello da seguire. Ciò significa che la maggior parte delle volte non sai esattamente cosa stai cercando o come la troverai. Anzi, non sai nemmeno se lo troverai o quale applicazione potrà avere un giorno. Eppure cerchi. Sei una che cerca. Kutato. So, you are a kutato.
Yeah.
And my question is not about the term kutato. It's about the importance of the family in the life of a kutato.
Oh, I was just ready to say, you know, in English, you know, so kutato means search, you know, and my father said I search for money in his pocket. But anyway, so in English, you know, we are not searcher, we are researcher because we have to repeat all of the experiment we are doing. We are re. And we always have to do one more experiment. That's if you read the book, you know that I have some Italian connection because Colombo was originally from Italy. So I watched this film, and that's my favorite.
Tenente Colombo, because we have also Cristoforo Colombo.
Oh,
but that's another one.
No.
So, il Tenente Colombo è il suo. But we have to define which Colombo. In Italy, you know, we are sensitive for the Colombos. But you are mentioning Lieutenant Colombo.
Yeah,
Tenente
the
Colombo.
Investigator. Yes, exactly. And so because you know, we always look for clues, and we always have one more question. If you are working in the laboratory, it's never ending because you have one more question, and you try to answer, and you get 10 more questions. Anyway, so the question is
The question goes towards the role of family, because
yes,
we started with your father, but then in the whole book, you speak about this dualism of being a scientist and the mother, a scientist and the daughter, a scientist and the wife. And it comes over and over, and you have many recommendations for women in science.
Yeah.
Can you share them with us?
Yeah, probably, you know, ladies here already you know, the forever young one at my age, you know, they know that. So we try to be perfect, yeah, perfect wife, perfect mother, you know, and then at one point we just forget it, yeah, because if you are a researcher or maybe physician, this, you know, family, work, life, let's forget it. There is no balance here because we you know, yeah, that we are scientists, and so that's. I try to, you know, I try to explain to myself, and then say always, you know, that the most important thing is that I am happy, yeah. And so then I can, you know, just like the aeroplane. You remember that the mask is coming down. First, you have to be happy so that you have the mask. And then you have your child, and you know you can help others. So that I always concluded I have to be happy. And then yes, so that with my daughter. So that you know, when I was practicing running, you know, I run the marathon. Did I mention? Yeah, 4.5 hours. 4 hours, 30 minutes I did at age 45. And, you know, my daughter was, have to eat quality time. So I was running and she was on the bike and saying, carrying the water to, you know, in the bottle, and then telling, Mom, come on, you can do it, and things like that. And then she volunteered also, you know, giving out the water at the race, and she didn't give it to anybody, you know, waited for me. By the time I'm coming, that Mom will get the. So, you know, that, and then you show your own example, that you come to save the lives of the patient, and that's, the child will learn more. That's, I try to explain to myself, to how we behaved. And then, you know, I'm not watching the TV and tell my daughter that do your homework, you know, that she, with our example, we showed the child, and then she will follow. And, you know, she decided, and, you know, she did different sports, and in and she was in college second year in sophomore when she started to row. And when people, you know, all of a sudden I became known, they said, oh, you know, you were not known. I said, oh, all of the US rower knew that I am Susan's mom. I am known. And when we went there, you know, they introduced me always that Susan's mom. And now she's coming, she's Scotty's daughter, yeah? And that's how we acted. So that's what I thought always, that with our example. And when I spend time with my family, it is, you know, quality time. Husband would be here, he would say, no, don't. But, you know, you have to, if you are, the young one is there, you know, you have to make sure that you find the right guy, yeah, that is okay, handsome, because the Italian guy is very handsome, but it should be also that somebody who value your dreams, you know, that's important. And guys, I have to say, you have to be that kind of husband, yeah, to value. And you know, and all of this life is just, you have to take easy, you know. I'm not just fun here. I am, you know, several picture was like 3 pictures in my life was taken in the lab, and I was not smiling for the camera. I was happy. So that you have to enjoy what you are doing and believe. You know that I go. I didn't show here that I grew up in a adobe house. In one room was my sister, my parents, 4 of us lived there, and you know, I'm going from there to Ivy League school. Even who's cleaning the lab is perfect English, and I, you know, 18 years old, I learned that this is the ceiling, so my English is not good. But I have to believe that with hard work, I can think something about what they don't. Those people, not who is cleaning, but those people who publish Nature Science, you know, and very smart people in Ivy League school, and so I had to believe, and that's what I tell all of the young ones, that you have to believe in yourself. And because, you know, we tend, especially women, that, okay, you know, the child, you have to help your elderly parents. Always everybody looks at the woman that should do something, you know, that, and we do try to do all of these things, but, you know, it's, together with partners to share, you know, the work responsibility. And so that's, I try to be a good daughter to my mom also, and good sister to my sister, and so, but we cannot be perfect. And don't, the young one I'm saying, don't even try to be perfect. And, you know, that's how it is.
Well, thank you. And actually, trying to be good, this is, here, actually, there is a number of young scientists, some number of senior scientists, and trying to be good each other. I would like to hear what you think regarding the relationship with the evaluation, because actually science is a matter of competition at the end of the day, and and things are changing. The way the work of researchers is evaluated is changing, and what is these times and in the next future, the best way is not only a matter of bibliometrics and what, again, is the relationship that the young researchers should have with the evaluation they will receive, and at the other way around, the role of the seniors.
I don't know what the question was, but I can say to you that when I work at the academia, you know, everybody's competing, and they wouldn't even start a project. I will be the first authors. You know, they already fighting over that. And then you go to the company, and we have to work together, everybody. There is no first, second, and everybody pull together, because if we don't come up with something which is helpful for somebody, then we all go home. We will go home, and it's somehow, you know, it doesn't discounting the references and publication. I mean, this is listen, my friend Csaba Szabó was just writing a book and reading in that one that, you know, the scientists get for review some horrible paper. He's reading, oh my God, this is terrible. Oh, they reference me. Oh. Oh, second. Oh, 5 reference. Okay, I will accept. So it is so slippery, you know, because I can collect all of these references. So that's how it happens, that, you know, they compromise. And then you have a number to measure, and it is meaningless. When I was in Leopoldina, was accepted to the Leopoldina Society, and who nominated me mentioned that they criticized I never published 100 papers. So if you not be happy, because more paper is about me than I ever published myself. And so, you know, they said, you know, usually 400 or 500 publication when we get somebody in, you know, the Leopoldina, not like less than 100, but he said that I convinced them that those 1-2 papers is there, and that's it. So you know, these numbers, which originally was some meaningful, it became meaningless, because this is the goal. I have to say that I usually say that we are started be scientists because we want to understand part of the nature, okay? And that was the 19th century. I had a nice cartoon. 19th century scientists wanted to understand, you know, nature, and the 21st century scientists want to publish in nature. So that if you want to understand nature and part of it, and then you have something discovered, you publish. And when you publish, then you write a proposal, because you want to have a team, more people would work on it, and then you get the money. I never get that part. I always went back and doing more on understanding nature. But then you get the money, and then you have more people. Now that students are coming, but yeah, you have to get your PhD, isn't that? And for the PhD, we have to publish. So you are publishing now that what is not ready, and nobody cares, but you have to graduate. And then, yeah, the boss want to be promoted, you want to be tenured, and so on. And somehow the shift from understanding nature goes that you are promoting your own career. And then it comes the disappointment, because listen, when you want something which you don't control, understanding nature, you can learn different ways, and you in control. When you want to get an award or something, I don't know, in the Karolinska 50 professor vote, and then they don't vote for you, or they, you know, the promotion, and who's the chairman, and you won't be the chairman, or you won't be promoted, and then it comes the disappointment. So if you want to understand nature, what I mentioned several times, then even you are happy when somebody is publishing you are just working on. Oh, that's good. Now we know more about nature. Nobody says yes. Because everybody is disappointing. Oh, my promotion is out on the window because I was just wanted to publish in Nature and some big journal, and now they are publishing, you know, when you are scooped. If you want to understand nature, you would be happy seeing that, oh, other people did, and they get part of the result the same. Oh, that's good. So I was right about. But that's not how it works, unfortunately, and nobody's happy when reading. But only like person like me, you know.
I would like to close reading your end of your beautiful book.
Okay.
And vorrei concludere rivolgendomi agli scienziati attuali, futuri e potenziali, e a chiunque altro desideri contribuire al progresso dell'umanità. Mi rivolgo soprattutto a chi tra voi potrebbe non adattarsi allo status quo. Magari non assomigliate agli scienziati che si trovano sui vostri libri di testo, magari non avete ancora imparato una seconda lingua o la parlate con un forte accento straniero, magari siete cresciuti senza mai conoscere un solo scienziato o avete frequentato una scuola in cui nessuno ha sentito parlare, o non capite le regole invisibili che sembrano governare gran parte delle stanze del potere. Spero che soprattutto voi terrete a mente le mie parole. Un giorno qualunque, dopo essere stata dimessa da una clinica, stavo tornando a casa e mentre percorrevo le strade di Szeged in condizioni di salute pietose, ho avuto un'intuizione. Che io dessi o non dessi il mio contributo, a nessuno sarebbe mai importato un accidente. Nessuno avrebbe bussato alla mia porta implorandomi di continuare a lavorare. Se tutto a un tratto mi fossi fermata o se un po' alla volta avessi ridotto i miei sforzi e non fossi riuscita a esprimere tutto il mio potenziale, la perdita sarebbe passata del tutto inosservata agli occhi del mondo. E un mondo privato di un contributo importante sembra ordinario. È la definizione di status quo. Non so da dove venisse la mia intuizione di quel giorno, ma mi ha accompagnato per anni e anni, anni in cui ero come invisibile, anni in cui quasi ogni messaggio che ricevevo, implicito o esplicito, sembrava suggerire la stessa cosa. Questo lavoro non fa per te, Kati. L'intuizione mi ha aiutato ad andare avanti, mi ha reso testarda, laddove altrimenti avrei potuto arrendermi. Forse non vi capiterà mai di essere colti da un'illuminazione improvvisa come la mia. Allora voglio essere io a dirvelo, adesso. Non fermatevi. Il vostro contributo futuro potrebbe essere ancora ipotetico. Ecco, vi prego, trattatelo come se fosse reale. È importante anche se non riuscite a intravederne le possibili conseguenze. Quest'ultima parte di noi ha un potere, nessuno ha il potere di controllarla. Continuate semplicemente a fare come fa il tenente Colombo. Una cosa, un'altra cosa, un'altra cosa, un'altra cosa ancora e poi ancora e ancora. Una cosa che so per certo è questa. Ogni seme dà origine a nuova vita. Questa vita a sua volta produce nuovi semi, che a loro volta danno origine ad altri semi ancora e così via. Il succo del discorso è che dovete fidarvi di ciò che è dentro di voi. Coltivatelo quel qualcosa, anche quando, anzi soprattutto quando, nessun altro mostra interesse. Insomma, continuate ad andare avanti, continuate a crescere, continuate a muovervi verso la luce. Voi siete il potenziale, voi siete il seme.
I hope, I don't know, it was Italian, I just understand Colombo, but you know, if I hope that I convince you today that I am not different than you. I am one of you. You know, just a girl from Hungary and, you know, doing things. Just because I read this book, you know, about how to handle stress, I could manage things. And so that was the advantage. But you can read my book. I quote a lot from that book, and then you know you can live your life the same, everything happening, but you are much happier. Yeah.
Thank you so much. Thank you so much, Kari. It has been a fantastic afternoon here. I really appreciate you being here with us, and again, hope it will be the first of another number of seminars here. Grazie di essere stati qui, spero che vi sia piaciuto. It was unforgettable for me, really exciting, and the special person she is. Grazie.
Thank you.